So now we're going to talk about gene regulation in both prokaryotes and eukaryotes. For eukaryotic gene regulation, we have a DNA sequence, and on that we have a gene. Upstream of that gene, you have a regulatory sequence. And then you have a regulatory protein coming from a regulatory gene coming downstream of the DNA. This would allow for them to grab on to the regulatory sequence and for RNA polymerase to grab onto it. You also have transcription factors, which will also allow the RNA polymerase to bind to the regulatory sequence. It'll fold over and allow the RNA polymerase to activate the gene and produce proteins to mimic the cell function. For prokaryotic positive control, we're going to use the LAC operon. On the DNA sequence, we have multiple genes present, and one dealing with lactose where they can all be transcribed together. Then we have a regulatory sequence where RNA polymerase can grab onto, and then we have the operon and below it a repressor. The reason it's called positive control is because when lactose shows up, we want proteins to break it down. So when lactose is present, it's going to unbind the repressor from the operon and allow RNA polymerase to express the genes and create proteins to get rid of the lactose. And once all the lactose is gone, the repressor will bind back onto the operator and turn the off switch on the operon. For prokaryotic negative control, we're going to use the example of the TRP operon. We have all the same structures, such as the promoter, the operator, the repressor, and the gene sequence. The difference is that when tryptophan, the amino acid, is present, the operator is off because the amino acid is binding to the repressor, and it will block RNA polymerase from expressing the gene. But when TRP is gone, the repressor will unbind from the operator and allow RNA polymerase to express the genes to make protein. That way they will make more tryptophan, so it can bind back to the repressor and cling back to the operator so that it will turn the sequence off.
Today we're going to be going over transcription. The transcription is the process of DNA turned into RNA. So basically DNA carries the information that basically lets proteins be made and helps the cell just run its course. So DNA can't operate on its own, so they basically transfer into RNA to be that protein. So this is what transcription looks like for prokaryotes over here and eukaryotes over there. So for prokaryotes, it takes a place in the cytoplasm basically because there's no nucleus, and DNA turns straight into RNA and turns into protein later on, basically seamlessly. Whereas in eukaryotes, it takes place in the nucleus and it isn't immediately ready to go and turn into proteins right away. So here's that eukaryotic sequence. Basically, like I said, it's not ready. So the 5 and 3 caps at the end kind of keep that RNA secure as it continues to develop. And there are introns and extrons. So basically the exons need to be spliced together through splicing into like a solid exon, which get ready for protein synthesis later. So that's the eukaryotes slowing down, whereas prokaryotes just go straight through. So they need to be spliced together. So in prokaryotes, transcription looks a little different. So this is basically ribosomes joining on to an RNA strand that's just starting to form as it's in its beginning stages, and it's essentially all of the stages kind of going together. So that's translation kind of streamlining along with that process. And this last part is how the promoters mix for prokaryotes versus eukaryotes. So eukaryotes have like 3 different RNA polymers. So basically they have their promoter and then they go to a single gene. So they're polycistronic, whereas prokaryotes that are monocistronic have that one promoter for all of them. So like basically the first promoter is RNA, second promoter is mRNA, third promoter is tRNA for eukaryotes, whereas prokaryotes, you basically are just looking at, one promoter for all those RNAs. And then back here, you can see mutations sometimes where basically DNA will have weird variations and it'll drip down transcription-wise into RNA and eventually work into mutations with proteins and essentially just mess up how the cell continues to go.
Here we have prokaryotic translation. It begins with 3 sites. The E site, which is where the deactivated tRNA exits. The P site, which is where the growing chain is held. And the A site, which is the aminoacyl site. We have these 2 sequences, the 16S RNA sequence and the Shine-Delgado sequence, which is complementary. We start out with a small ribosomal subunit, which is the little boat-looking thing, which is disconnected from a larger subunit thanks to IF1 and IF3, which are shown right here. It binds to the Shine-Delgado sequence because it is purine-rich. Here comes the Shine-Delgado sequence. We then have tRNAs coming in with GTP and the initiation factor 2. It binds to the Shine-Delgado sequence before the GTP is converted to GDP. Once this happens, all the initiation factors leave. And then the large ribosomal subunit connects back. Another tRNA is brought in, but it does not have a matching sequence. Therefore, it is factored out, and a new one comes in until it matches. Once it matches, the sequence keeps moving along, leaving room in the A site for more tRNA to come in, until the kill factor happens, in which case a release factor will come in, and this is eukaryotic translation. We have an mRNA here. The EIF4E factor binds with the EIF4G factor and then the PABP factor on top of that. The poly A tail of the mRNA interacts with the PABP protein. Through this interaction, they bind, causing the mRNA to turn circular as shown. The 43S complex binds with the mRNA molecule at 5. It then scans for the AUG codon. The top half, or the 60S subunit, is recruited to the site, turning it into an 80S complex. From here, more tRNA will come in, creating a large chain until it is terminated by ERF1.